not too far from the hypothalamus. It's literally right underneath, and it signals that pituitary gland to release this adrenocorticotropic hormone called ACTH, and then this hormone travels down to the kidneys, all the way down to the kidneys, and this ACTH signals the adrenal glands to release cortisol. And cortisol is where everything kind of makes sense and happens, and it causes the changes in the body that helps deal with this stress. So the HPA axis is the most likely the underlying route between this connection. So this was one of the studies that I found, and it kind of focused on the effects of general gut composition on stress and anxiety levels in the stress axis and anxiety levels, and they observed rats. So they tested whether the absence of no gut microbiota had an effect on anxiety levels. And so they used germ-free rats, which germ-free is they haven't been infected by any microorganisms, so were born and raised in a sterile environment, compared to specific pathogen free rats. So these rats were tested for specific pathogenic microorganisms to see that they don't have it. So they used these two rats, and they examined their behavior using this maze called an open field, which a lot of these experimenters use this because it's with rats. They analyzed their behavior and social interactions throughout this open field test. When, like, 2 times whenever they were at adolescence, which is 30 days old, and then whenever they're at adulthood, which is just 71 days old. And then they focus more on like an analytical, like with the cortisol concentration, expression level of the CRF gene, and like in the hypothalamus and the glucocorticoid receptor gene. So that's kind of dealing with that HPA axis right there. Before they did start this experiment, though, to prevent some abnormalities in the results, they tested each rat for neurological behavior disorder or abnormalities that could skew the results at the end, and then they put them through this open field maze and recorded the results. And they ended up with that the germ-free, the GF rats, displayed greater anxiety-like behavior in the open field test compared to these SPF, or the specific pathogen-free rats. So they determined that the anxiety-like behavior included going to the center of the maze fewer times, an increased number of defecations, and then the time, the time between different actions increased, and then the time spent in corners, because they felt safer in the corners, increased as well. So as we can see, the germ-free mice displayed more anxiety-like behavior. And then the p-values for each of these were significant because they were below that 0.05. They also showed an increase in transcription of that CRF gene, a decrease in transcription of the GR gene, and this in total kind of intensifies that HPA axis, and so it intensifies the anxiety-like behavior because it's causing that HPA axis to increase, and like just the signals are being sent, and it's kind of going crazy. So also on this side, this is where it shows that these rats, there wasn't really a difference whenever there was no open field stress test between for the rats, but then whenever there was an open field stress test, there was a difference between each category, and especially with the gut germ-free mice, or rats. They didn't have any gut microbiota, so they had the increased anxiety-like behavior, and the increased neuroendocrine response to the acute stress. Another study I found was that gut composition modulation and their effects on anxiety levels. So these researchers, they really focus on the genetics of the mice and how it affects their anxiety levels because, like I said before, that genetics really plays a difference, really makes a difference. And so they observed 4 genetically different mice, and they measured their initial anxiety without any gut microbiota modulation, and then they measured their anxiety levels with gut microbiota modulation. Specifically, they used this probiotic. They delivered about 200 microliters twice a day for 4 weeks, and it was called Lactobacillus rhamnosus. And as we can see, there was a significant difference when it comes to the open arm duration. So as the weeks progressed, one of the species really, their anxiety-like behavior really decreased because they spent more time on that open arm, on that elevated or that open maze. For the other ones, they really didn't show that much of a difference. So in conclusion, the gut microbiome, there still needs to be so much more research done on it, like more experiments and more knowledge to be learned. But it is an intricate and extensive system that plays a vital role in many processes that occur throughout our body, but specifically with the connection between bacteria that resides in our gut and our brain. There is a connection for sure, and there is a connection between anxiety and our gut microbiome. Now, I'm not saying, and it's also this, but like the connection can be made because of this HPA axis that I stated earlier. I believe that there's always more experiments to be done, that in future studies they should probably do more studies on humans because they did a lot of these studies focus on rats and mice, and that's great, but they're, to a certain extent, we have different, we're a little bit more complex. The researchers probably could have given the mice and rats more acclimation time, and I don't want to say that this could have skewed the results, but it probably might have skewed the results in the end. Not all of the anxiety-like behavior could have been due to the absence of gut microbiota or the modulation of gut microbiota. And then probably in the future they might want to mock the actual environment of mice or rats. I just want to acknowledge my wonderful mentor, Sarah Colosimo. She was such a great help with this whole thing. I can't thank her enough. The University of North Florida Biology Department, my parents and friends, and Starbucks coffee, and the free Wi-Fi. Thank you for watching my presentation. Do you have any questions?